This research explores Geogemma barossi, also called strain 121, this little wild archaeon that basically lives at the limit of what life can tolerate. It was discovered back in 2003 at a deep-sea hydrothermal vent off the coast of Washington. A research team sent down a remotely operated submarine to a black smoker chimney. They grabbed a sample, and inside it was this little organism that can actually grow at 121 degrees Celsius. For reference, that's the temperature we use to sterilize lab equipment. Before this, scientists thought life topped out at 113 degrees Celsius, so it completely shifted what we thought was possible at extreme temperatures. The cell itself is tiny, about 1 micrometer, and it's coccoid, so it's kind of spherical. Like other Archaea, it has a membrane periplasm outer layer structure, and strain 121 also has a tuft of flagella on one side, which is called Lophotrichus flagella. Genetically, it sits deep on the archaeal branch of the tree of life, meaning it comes from one of the earliest diverging lineages. It even has a special enzyme called reverse gyrase that literally supercoils its DNA tighter so the strands won't melt apart in extreme heat. That's one of the key features that allows it to survive where almost nothing else can. Metabolically, it's an obligate, anaerobic chemolithoautotroph. So basically it needs zero oxygen, it loves super high heat and pressure, and it eats inorganic chemicals instead of organic food. It uses ferric iron as its electron acceptor, so iron is a key part of its metabolism. It grows between about 85 and 121 degrees Celsius, and it actually can't reproduce if it drops below that range. Its home is inside of black smoker chimneys. Some of these towers are 79 feet tall and reach temperatures near 300 degrees Celsius. They spew out superheated water loaded with dissolved iron and sulfur. So when the hot water hits the cold ocean water, all of those minerals precipitate out and create dark, smoky plumes, which gives them their name. Because of the extreme environment, there aren't many organisms around, so Geogemma barossi lives in a pretty sparse community. We don't know much about its competitors or its partners, but it probably crosses paths with other hyperthermophiles and possibly archaeal viruses that hang out in the vent systems. As I learned more about Geogemma barossi and its environment, I started translating those elements into my ceramic work. My vessel reflects the biology and geology of hydrothermal vents through clay, glaze, and form to echo the conditions in which Geogemma barossi exists. Movement was super important for this project, so I created movement by carefully shaping a simple cylinder into a form inspired by the flow of superheated water rising from a hydrothermal vent. The black base color represents the total darkness of the deep ocean where these vents exist. And the crawling texture creates a rough, uneven surface, while the beading and dotted glazes create clustered forms, and that's similar to the way Geogemma barossi grows on hydrothermal vents. These textures hint at small structures forming beside one another over time, the way microbial colonies and mineral deposits build up in close, irregular patterns. The color palette I chose to use comes from the chemistry of vent water. Black is from iron sulfides, the green is from chlorite, yellow from sulfur, and there's bright orange, flashes of orange from iron oxides. In the kiln, these glazes melted, split, and pooled in their own way, almost like chemistry happening around Geogemma barossi in real life. The vessel isn't meant to look exactly like the organism, but to capture the feeling of heat, texture, and constant motion. The whole piece is my interpretation of Geogemma borasi, a microscopic organism living right at the edge of what's possible for life and the extreme environment it calls home.
Hi, I chose to write my project on methanogens and how they impact the world. Hi, I'm Manny, a methanogen. My family belongs to the Archaea domain, and we come in many different shapes and sizes, from spherical shapes called cocci to long and short rods called bacilli, even irregular shapes. Since there's so many of us, we have to specify that my genus is Methanobrevibacter and my species is Ruminatium. I'm anaerobic and I prefer to only be with close friends. You'll usually find me all alone in a pair, although as I get older, you'll find me linking in very short chains of bacilli. Some may call me lazy because I'm non-motile and non-spore forming. But I like to say I just use my time more wisely than some of my relatives. I am a gram positive. Many of you have siblings and cousins that make up your family tree because they're genetically similar to you, but I have subclusters instead. Researchers use our 16S rRNA gene sequencing to identify us. I like to think of my first subcluster as siblings. There are 9 of them, and they are 98% similar to me. And a second subcluster I like to call cousins. There are 15 of them, and they're 97% similar to me. We are all very similar to other methanobrevibacters, although I require a coenzyme M to undergo my biological processes that others don't. While your DNA is a double-stranded helix, I only need a single-stranded circular chromosome to complete all my metabolic functions. My family and I can live in vastly different oxygen-poor environments, such as wetlands, landfills, deep-sea sediments, and the Earth's crust, as well as the digestive tract of other mammals and insects. I specifically prefer a more well-balanced diet living in the rumen of livestock. Researchers have tested when I began colonizing by using a syringe and stomach tube aspiration of newborn lambs. It showed that I colonized at birth and before the full development of the rumen of livestock, and I begin to slowly grow in population while livestock are nursing on only milk, although my population increases exponentially with the full development of the rumen of the livestock and when a grass diet begins. All organisms have different metabolic processes that convert fuel sources into energy. You eat food to get the energy you need to grow, while I live in cows that eat grass or grain. I must make friends with many different bacteria to complete my metabolic processes. I form a symbiotic relationship with fermentative bacteria in the rumen of the cattle because they undergo enteric fermentation to break down the cellulose in the plant's walls and produce carbon dioxide and hydrogen. After the bacteria produce CO2 and hydrogen as byproducts, I use them as substrates for hydrogenotrophic methanogenesis, converting the carbon dioxide and hydrogen into methane. Methanogenesis converts hydrogen peroxide into methane using that hydrogen as an electron donor. You see, you produce carbon dioxide every time you breathe, which increases the world's entropy, and I reduce the amount of this greenhouse gas in the atmosphere. This might make you think that I'm a bit of a hero. However, Stanford's study made me into a villain when it released a study showing that the methane that I produce is far more effective at trapping heat in the short term than carbon dioxide is, and is actually 80% more potent, proving the methane I produce is actually increasing global warming. Global warming can affect you and future generations by causing more wildfires, heat waves, coral reef bleaching, and even severe droughts. There has been promising studies showing that adding only 2% of a red seaweed from the Asparagopsis genus to feed cows and to cow feed can actually reduce methane production by 70% without altering the fermentation process. It does this all by binding to the active site of coenzyme M and inhibiting the final step in methane production. All organisms have both positive and negative effects on the universe, and I hope that hearing my stories has helped you gain more insight into methanogens while highlighting the importance of sustainable sourced meat.
Hey guys, I'm here to talk about Halobacterium salinarum today. So let's get started. So Halobacterium salinarum is an archaeon which thrives in high salinity environments, making it a halophile. It will only actually start to grow when the concentrations are 3 times higher than salt water and thrive in concentrations 10 times more than seawater. They're able to withstand high doses of radiation and are able to survive for thousands of years entombed in salt. They can be found in salt lakes, solar evaporation ponds, and colonize in salted foods. And their signature hues of pinks and purples make them easily recognizable. So the H. salinarum is rod-shaped and motile, and archaeum is the flagella that's used as the swimming device, which is made up of sulfated pentasaccharide units and a little-known second glycan. And the flagella allows the archaeon to move to depths for maximum light absorption. And instead of a cell wall, it has a single lipid bilayer surrounded by an S layer of glycoproteins making up the protein shell. And the reason why it needs to get to a higher surface is so that the protein cell can use photosynthesis, and we'll get more into that later. But the carotenoids in the cell membrane are responsible for the red or pink colors that are expressed by this archaeon. H. salinarum is from the phylum Euryarchaeota, and their order is Halobacterialis. There are 8 genera: the Halobacterium, Halobaculum, Halogeometricum, Halomicrobium, Halorhabdus, Halosimplex, Natron bacterium, and Natronomus. And they're halophiles related to a diverse group of chemoheterotrophs that grow also in a variety of extreme environments. The phenotypic data is limited, but research shows that they are a monophyletic group. Halobacterium salinarum are facultative anaerobes who utilize aerobic respiration, photosynthesis, and fermentation. H. salinarum oxidizes pyruvate, which goes through the tricarboxylic acid cycle using pyruvate ferredoxin. The glycoproteins in the H. salinarum plasma membrane are able to convert light into energy. Bacteriorhodopsin is a transmembrane protein that serves as a plasma pump that uses light photons to pump hydrogen ions across the membrane to create a proton gradient. The alpha helices of the Bacteriorhodopsin contains 4 retinol proteins that have pigments involved in the light energy conversion, which those are what contributes to the different hues of pigments that we see in this archaeon. The proton gradient produced by the hydrogen ions across the plasma membrane is used by ATP synthase to generate ATP. Halorhodopsin is a light drive chloride pump in the membrane that helps maintain the salt concentration internally during growth. The sensory rhodopsin 1 mediates the photolytic response to orange and UV lights. Sensory rhodopsin 2 is involved in phototaxis that mediate the photophobic response to blue light. In the absence of light, they are able to grow anaerobically in the dark by fermenting arginine to ornithine, ammonia, and carbon dioxide. The environment that H. salinarum grows in is hypersaline, which typically has a high level of radiation when drying up. It has adapted to this environment by having a high cellular concentration of peptides, phosphates, and manganese to protect the cellular proteins. This allows H. salinarum to withstand high levels of radiation. Enzymes are able to repair the damaged nucleic acids and have multiple copies of the chromosome for repairing DNA. Dunaliella salina is a green alga photosynthetic microbe that can be found living alongside the H. salinarum. D. salina has a glycerol-filled cytoplasm to create the osmotic balance, Glycerol from the desalina is a carbon and energy source for H. salinarum, and symbiotically, H. salinarum provides nutrients to stimulate growth of the alga. When entombed in salt crystals, H. salinarum are able to obtain organic matter from co-entombed microbial cells like the desalina and other dead cells of the halo archaea, following H. salinarum to stay alive. Well, thank you for taking this tour of H. salinarum with me. I hope that it stays entombed in your memories.